Welcome everyone to the last session of the day. So I have the pleasure of introducing Jana Kholova. Well, Jana has a lot of experience on phenotyping and applying these phenotypic methods to the crop growth modeling context. She's a researcher at the CGIAR in ICRISAT and also at the Faculty of the
Czech University of Life Sciences.
Sorry, I was
It's fine.
So the floor is yours. Welcome, Jana.
Yeah, you look still alive. Hope you had enough of coffee so that you are ready to what is coming up for you. So we are going to have a little bit deeper dive on what is happening at CG institutions regarding modeling approach and to analyze G by E by M, and then to serve certain type of end users. And then I hope it works. Doesn't. Should. Yeah, so structure of this presentation is as follows. So little reminder of CGIAR institutions and what ICRISAT is within that structure. I was lucky that, that Martin already did most of the job there. Then general principles, again, I will not need to remind the community in depth about it, but still we will look at the basics of what kind of tools we are using and how do we want to, what are the mechanisms which actually bring the value to the end users. And we will look, maybe we will use more time to look at the case studies. Both of them are breeding. So one is the peanut breeding team and how this approach is, which I'm going to present, has helped, hopefully, the peanut breeding team in South Asia. And then we will look together on what is happening with sorghum in West and Central Africa. So, 1CG, so that institution stands for Global Research Partnership for a food-secure future dedicated to transforming food, land, and water systems in a climate crisis. So this is the conglomerate of several institutions with different mandate. ICRISAT, International Crop Research Institute for Semi-Arid Tropics, is particularly concerned about the dryland farming systems in South Asia and across African drylands. And there goes even appropriately the mandate. So these crops, which are mandated to ICRISAT, are the poor man's crops. We are basically looking at the most complex systems with the most risky environments and poverty-threatened farming systems. So we work primarily at the crops which are at home in those regions and which provide livelihood to those farming systems. So that is sorghum, pearl millet, finger millet, peanut, chickpea, pigeon pea. But of course, we work beyond that too. So let's look in a brief to the general principles, how the crop growth models actually help us to generate the value for the wealth of data, which we are now gathering. So this is something which I don't need to remind you about. It's just the reminder that we are using the mechanistic crop growth models, which helped us to basically transit from the static information about the systems to the dynamic one, which now can be used to understand and design the systems much more effectively. And you see that we have been quite active in that space. So what type of information we can get from this approach, which actually helps our end users? So the primary goal is to analyze the systems, to look at the heterogeneity of the main crop production systems, and also system drivers and limitations. So this is the baseline of the digitalization approach. And the next step is much more, let's say, data intense. So there we are trying to test various what-if scenarios and look at the particular combination of genetics and management options at across the spatial temporal scales to figure out what is the optimal designs of the system, and of course the foresight, which many of the colleagues were talking before. So for whom we work? It was quite interesting that I didn't hear much about for whom we work, for whom we are developing all these approaches. So at our end, or at least most of the use cases goes to the support of the breeding teams at ICRISAT. So what are the questions of these breeding teams usually? So they are looking at the optimization of the production systems. They are looking at how to make systematic and more effective way to test their crops in multi-locations, especially when the resources are limited. I'm allowed to say that, hopefully. Then their typical question leads to what is the ideotype, how the crops should look like if they are to cope with current and future scenarios. Of course, that helps us phenotypers to design the phenotyping protocols much more precisely, accurately, and targeting manner. And geneticists, of course, they have to dig deep on what is the underlying genetics of those ideotypes. Of course, foresight is something which is particularly enabled by these approaches in a much better way than any others. So, of course, we do have a power to assist also agronomic research and the primary producers. We are not quite there yet, but these protocols can be also equally useful for optimization of the agronomic trials and, of course, decision support as a decision support tool to primary producers. So we have been quite busy digitalizing all those systems for sorghum production, pearl millet, chickpea, peanuts in South Asia and in African lands. So these exercises range from few gigabytes of data, let's say, in initial trials, and Greg might be laughing now. Just last week, we finished producing 20 terabytes for the design, to support the design of the sorghum production systems in West and Central Africa. So that's where we are right now. It's not really fun for the biologists to deal with this kind of heaps of the data, especially when we know that what really creates value for the end users out of those terabytes is a tiny fraction of it. And I do see more and more as I keep going from conference to conference that people actually struggle in this space. We generate too much, but the value for the end user is really the very little information out of those heaps. So now the trick is how to find it. So here we are organizing ourselves a bit more effectively in that space. So we are forming the platform to support this kind of digital efforts in the agriculture space. As far, we have identified 3 domains which are the common denominator to do all these efforts. So of course, we need very strong data management systems. Surprisingly, nobody talked about the FAIR principles, about the digital responsibility goals, about the ontologies, about the database. It's very boring stuff, but absolutely necessary if we want to scale up, or at least from our experience when we want to scale up our efforts. Of course, people talked about the phenotyping, so computer vision specialists, how to extract features which then can provide the value to breeders, for example, is absolutely key. And how then to extrapolate all this knowledge across time and space. So that's the domain which I would like to talk more about today, is agroeconomic system modeling. And we do have a team looking at this one intensively. So if this resonates with you, let's join the hands. We would be particularly interested to work with you in the topics which lead to the sustainable agriculture goals, as Martin was talking about in the morning session. Deep breath, let's talk about peanuts in Asia. First case study, hopefully successful one. So this is the work of Amir. I don't know where is he, but that story started long back, published in 2021. So this is what you are looking at is system analysis for the major peanut production tract in India. So this has been done on demand of peanut breeding team. So they were particularly interested what are the major production zones and how diverse they are. So this is what you see on this map. We call it TPE, Target Production Environments. So those are the colorful patches. And the different color here means that those different regions basically are more homogeneous within the region, while more heterogeneous between those colors. So you see that it's quite different for what breeders have been using so far. And hopefully we are bringing more value to them by giving more information about these systems now. How is it used? So of course, we don't have endless resources. So this is now used in practice to systematically test and mainly prioritize where the breeding team wants to test their new cultivars or supposedly new cultivars. So they are based on those production characteristics and size. They are now going for prioritization of the several of those TPs over the others to maximize efficiency and resources. So that was still possible to run in the laptop. Now, when we look at the system design exercise, when we want to test range of genotypes, range of environments, range of management practices, then it goes poof. So this is some slide which I got from our IT team because apparently you computer scientists are getting excited about these numbers. So we tested, I don't know whether I would say it right, but this is the information which I got. So we tested around 100,000 of those combinations for more than 1,000 locations in India. So we needed quite powerful machines. It took 2 days to process it. We successfully generated around 2 terabytes of data, and this is something which apparently is of matter here. I have no clue what it is, but apparently this is this is what matters in this slide. And of course, we also cannot do it just like that, no? So our team was quite efficient in designing the modular tools where they can do some analytics on run and swap the modules based on what model we want to use, what kind of statistics we want to plug in. Well, I would like to know your feedback on this particular slide. If you have something better and more efficient, then I am all ears. So now, 2 terabytes of data, what do we do with this? So we are trying to look at the adaptation landscapes and to figure out what is the optimal genotype and management combinations for those target production and environments in multidimensional space. And what really helped us there a lot to figure out what the priorities is to look at what the end users actually need. So that stratified quite a lot to figure out what kind of parameters and what kind of risk measures we have to involve in the analytics. The result is this beautiful little map, which shows basically the adaptation zones for peanut production in India. So this one little colorful map is all the summary of those 2 terabytes of data, which now is being used to define the peanut ideotypes. So I have very much simplified the outputs, how it could look like. So for example, what we are looking at in this little table on the right side is the output of the system design analytics and the target phenotype for this particular cluster, which is the desert zone of Western Rajasthan for irrigated and for rain-fed systems. And here are our beloved traits, so some which are related to vigor, some which are related to water-saving mechanisms of the crops. And what you see, these numbers, is the importance of traits for stabilization of production in those regions, in that particular cluster 1. So you see that vigor is quite important, but what I want to drive your attention here is this. So if we are in irrigated systems, the importance of those water-saving traits is maybe not so much, but the moment we are moving to the rain-fed system, then the importance of those traits, which help plants to use the water resources more efficiently, suddenly raises up. So this number is the indicator which tells us how much we destabilize the system, provided that breeders do not take these important traits in account. So this is already the quantification of impact or potential impact of all those traits which we all talk about and like to analyze. So of course, now phenotyping, how do we phenotype for it? So there is now not a single answer to this, but for us, the entry point into the breeding populations is this system, LYSISCAN, which is able to cater with very high numbers of lines in a few week assays, measuring exactly those traits, which we modeled in our system design exercise. So basically that modeling exercise gives us the recipe, which then we can execute in a breeding pipeline using these technological tools. So but as I am saying, it's not, there is nowhere a single solution for phenotyping when it comes to the breeding pipelines, at least that is our experience. So this is very simplified scheme of the peanut breeding pipeline. So you have some crosses, of course, and you have some filial populations where at some point you are dealing with thousands of lines. So in the first part of the breeding pipeline, we are basically looking at the technologies which are helping us to saturate the populations with the climate smart features. And in the second, we are looking at how we actually accelerate the post-harvest quality using various types of different technologies. But again, these are very nice tools, but how do we actually measure whether we are making the process better or worse? So in this case, we are using the metrics related to the genetic gains, which already we talked about. Plus, what is really important is the time and cost of those evaluations. So in the first part, and there are details about it, we are actually addressing the selection accuracy for those climate smart traits, let's say. In the second part, we are reducing the time and cost of post-harvest evaluations. I didn't really dare to put the numbers on this slide, but it looks like that within the generation, we are able to save around $25,000 using this technology accelerated approach. So now I have very little time to look at the sorghum case in Africa, but I will try to be extremely quick. So when we try to implement the same approach, we are stuck at the first step because models are not doing what we want them to do for the type of materials which is typical there. So there, I really have to thank the UQ team, which helped us to enable certain mechanistics within those models so that we can now reliably model the indigenous sorghum. Same data. A huge problem with the data availability. But anyways, we succeeded with some basic environmental characterization for Mali, at least. Now we are extrapolating this system design exercise for full West and Central African regions. You see that it's, again, huge data intense exercise, and I cannot wait to get back home and start looking and digging into the details, what the genetics and what the management would suit those particular lands. So just want to drive attention to few of these constraints. Everyone is maybe presenting here the stuff in rosy pictures, but unless we give a name to constraints, less is what we can move. So definitely the crop growth models helped us to understand the environmental context, the relevance of particular genetics for those particular environmental clusters. Data availability remains a big problem in the systems which we work for. Data availability and quality, both. Management and utilization, we are getting organized in that space. Transferability of tools and methods, big problem. We cannot just take one system from Europe and apply it to Africa. That would be disaster. And Martin also talked about community development and networking, and that's where I'm so happy to be here with you guys. So thanks so much for inviting me to share with you this story. So in summary, data to value, I want to stress it. I think we are very efficient as the community generating all those data, but what creates value for particular end users is very little, and figuring that out is not easy. Relevant tools, methods, and technology bundles, I hope I was clear about it. Measuring the impact, absolutely necessary. If we want to implement new tools, we need to make sure that we are not actually doing worse than was the original process. That was our big learning. And community and networks, yeah, it's always about the people in the end and their wills and skills. So with this, I thank to all our amazing partners, because alone we are nothing. We are just the network of people with a goodwill who are trying to solve some of the burning, globally burning issues. So thanks so much for attention.
Thanks, Diana, for the nice presentation, for staying perfectly on time. So we have time for one or two quick questions.
Everyone is speechless. Good.
Okay, thanks. It's nice to have someone that can have good throwing skills for the microphone.
Thank you for this very nice talk. You mentioned that data availability is a huge problem. So maybe you said it, but I missed it. For the India case study, what was your data source for the idiotyping part, for the training the model? Especially the soil data is always a huge problem, isn't it? So did you use a soil database, or was it measured data? Could you elaborate again?
Brilliant question. So you know what is the very useful Czech trait? I am from Czech Republic, so we people are able to make something out of nothing. So of course, so we had to be quite creative, but the best data set on soil came from the University of Queensland on India. And thanks so much, Greg, again, and all the UQ folks that you didn't leave us alone in that struggle.
One more question? Okay.
Short one. Are your breeders collecting multi-locational data that you can use to evaluate your crop models? Because I think that's an important component.
Mark, please, can you answer that? I want to be politically correct, so I would rather not answer that question.
So at the moment, I think the answer is definitely no, but they can. Is that what you would want to say, Jana?
Thank you. More question by dinner.
Yeah, we have 1 minute because we are waiting for the microphone. So if there is one last question, we can make use of the time before we give this floor to Sinu.
Oh. Okay. So yeah, you showed all of these environmental data, but I was wondering if you also have some insights on what is the genetic part that is beneficial. So did you analyze more like genomic regions or things like that?
Time to show it, but we do have a lot of, let's say, work in that space. So something is related to what Janila was talking about the stay green, for example, in sorghum. Maybe I can give you some of the publications and we can discuss more about it in details later.
Thanks.